Okay, should we get going? So good morning, everybody. Welcome to the focus session from this project called European Bank for Induced Pluripotent Stem Cells, or EBiSC. My name is Tim Allsopp. I am affiliated to Pfizer, and Pfizer is the project leader of EBiSC. So just I'm going to give you a few words of background to the context of the project, and then I think really I'll hand over to my colleagues, project partner representatives in the EBiSC project, and give them the opportunity actually to tell you more detail about the different working components of the project. And really the key objectives from this focus session are just two. So the first is to raise your awareness about this big European initiative, big project ongoing currently in Europe, multi-partner project. And then also principally for you to actually take the opportunity to engage and ask questions and raise points for discussion amongst us all, but then primarily with the various project partner representatives in the room. Because what we principally would like to know from you in terms of potential IPS research grade stem cell line users is what you would like to see from this particular project in terms of the cell line catalog, research services, protocols, methodologies, etc. And without further ado, I'll just move on. Ta-da! Okay. So first important slide really about the context for the Innovative Medicines Initiative, which is the principal sponsor for this big European collaboration. So the IMI is the world's largest global public-private partnership working in the life science R&D sector. And for quite some years now, the IMI have been funding both preclinical as well as clinical stage development projects involving consortia constituted by both the private sector, so the pharma industry, as well as the public sector. And really the target of these projects is to focus on a research program that relates to a specific research challenge, principally posed by the industry partners in the project, with a view then to working through a project plan to come up with a solution. So the idea is that these types of R&D projects are to address key needs which are common to principally the industry, the stakeholders, the partners in the project, but then also principally the patients. So these projects have been running for quite some years now. In the sort of schematic on the right there, you can see from the first phase of IMI, actually the relative financial scale of the overall program, and the program funds many projects in terms of billions of euros from both the public and the private sector. And the scope from these projects covers all elements, principal elements, from basic research all the way through to pharmacovigilance. And they are founded on common principles of a pre-competitive nature of research, and typically they will involve an open innovation component. So a little bit of a history lesson. So in 2012, actually, Pfizer, being the principal party interested in addressing the research challenge, came up with a call topic. Pfizer then pushed this call topic through the RMI program in partnership with many other pharmaceutical companies who today are still working within the project. New ones have also been added during the project duration. And we got through the peer review process, and in 1st of January 2014, the project launched. And initially it was given support by way of a grant from the public sector, the IMI, to focus initially on demonstrating the foundation of a workflow, the process, so the core machinery of the solution for the problem. And the problem I'll come to in a minute. And we've been working through this now within a 3-year timescale, and we have a focus to actually extend the project beyond the current end of this year into future years. So the particular research challenge that we posed was that we ourselves in Pfizer and the industry per se had a particular challenge in terms of accessing standardized research grade reagents for disease modeling, iPSCs for disease modeling. And so we proposed actually that what would benefit Europe would be to have a centralized, harmonized process, even a facility that would allow operations at industrial scale in providing these research grade disease models and the tools for the disease models to the all sectors of the research community. So to set up a storage and distribution center with a catalog that would sort of encompass really all the principal research interests on an evolving basis in terms of what the research community was looking for in terms of disease models, quality of lines, etc., and to actually provide a full electronic commerce portal to enable, straightforward, simplified way in which third-party research users may access through the project the associated qualified cell lines in the catalog and any associated data. For the cell lines in the catalog to support diverse applications, so translation of research protocols to develop an assay for disease modeling, potentially extending on from disease modeling into a more sort of precision medicine, stratified way of understanding individual clinical phenotypes better within a single disease diagnosis, and also using these research tools for safety pharmacology and toxicology testing, etc. And then also critically to be able to set up the infrastructure and to have an operational business model and an operational plan, which eventually would lead to a facility that would be self-sustaining in terms of operations and self-sustaining on a not-for-profit basis. So really, again, just to reiterate, meeting demands generated, or meeting this demand actually generates revenue. This revenue can be used then to cover fixed operating costs of the operation, the workflow, that the industry contribution, the public contribution from the pharma company adds substantial value in terms of testing these cell lines in the catalog. And the idea is to actually bridge and formulate a contemporary evolving business plan that actually hopefully will fulfill the self-sustainability as part of the original objective. So in simple terms, the project outline, what does EBiSC plan to do? So I think ultimately the output of this will be to improve the European landscape, but of course ultimately also in terms of a larger picture, the bigger vision in terms of an international cooperation between IPS banking initiatives in various geographical territories. Then it's all about improving the research landscape. So if you imagine the simplified workflow, you have research projects which are busy creating IPS lines. EBiSC will sit in the middle between these projects which act as the depositors of the cell lines and other research groups who want to access these unique reagents, and they don't want to spend their research dollars on making similar research reagents of a similar grade, similar disease classifications, etc., then making them available to those researchers as third parties, and EBiSC sits in the middle. So the project has a number of core components. So first of all, we've established a central facility. It's based in Babraham in the Cambridge Biomedical Research Cluster in the UK, which is using best cell culture technologies to initially operate a sort of fledgling workflow, but then the idea is to have this scalable so that we can automate elements of it and be able to do this in a sort of much more industrial scale. We've built a data management system, which has a key element of interoperability, enabling different databases and information to flow within the project so that this information management system can then, triage and provide those elements of data associated with the cell lines in the catalog in a controlled way to third-party users. We are building the catalog. We will be building the catalog much more this year and into subsequent years. Of cell lines which are driven, again, in design by meeting what researchers need. We've actually gone back and demonstrated how we can harmonize and standardize the whole procurement element of actually obtaining biosamples through informed consent, anonymized informed consent, and to be able to put in place the deposition access user agreements which actually fit in a harmonized governance and ethics framework, critically associated in making the ultimate stem cell lines that are created from those biosamples, plus any associated data available within the project to partners and then also to third parties. We have common standards and testing of the cell lines in terms of qualification. You'll hear a lot more about that. And then also we have a work package component which is on a feasible, manageable way, selecting individual cell lines and carrying out more extensive cellular phenotyping of those cell lines in terms of disease modeling. And this is an activity principally driven by the industrial partners in the project. So what you're going to hear about today is hopefully some element that indicates to you that those bottom three components represent some movement beyond the art status in terms of making, banking, distributing, qualifying and distributing IPS lines for research. You'll hear from Julie Holder, who's going to tell you about how the catalogue is being built, what the ambitions are for future in terms of catalogue components, and then also talk more around the ethics and governance element in terms of consenting and contracts. You will hear from Laura Clark of the European Bioinformatics Institute about how the data management system has been constructed and how that operates, and you'll probably have a live demo as well, I understand, on that. And then finally, you'll hear from Glyn Stacey around how we've established this QC platform that meets international standards in terms of qualifying cell lines and making them available to the research community. One snapshot on project outline. This is the consortium at a glance. We have many partners spanning the private and public sector. We have non-government organizations. We have the Wellcome Trust, Sanger Institute, the world's largest charitable organization. We have a very discrete, closely fitting cluster of biotech companies, as well as some world-famous research institutes busy making IPS lines and using them, etc. Okay. So just to leave you with this, so this slide really just summarized latest information in terms of how you can learn during this session and obviously subsequent to this session more about the project. We're going to have this an initial sort of show and tell element, and then we'll have a break, and then we're going to enter into a panel discussion. And there are two discussants who we've drawn in to actually give to you some element, three actually, Julie's just reminded me, discussants who will present to you actually how they've actually initially collaborated with the project. So they will give you some initial background on who they are and what they're interested in, and then also hopefully describe what their interests are in the future in terms of working with the EBiSC project. And in this panel discussion also there'll be the opportunity for you as interested parties potentially to put your hand up, raise questions, and hopefully engage in discussion. Okay? So that's a brief snapshot by way of introduction to the project. Gives you an overview actually of what's coming next after the break. And without further ado, I'll hand on then to Julie, who's going to tell you more about the real elements of what the project is doing. Thank you.